Welcome. I want you to imagine a scenario that, well, it sounds a bit like a paradox.
Yeah.
Picture a heart that squeezes perfectly, like powerfully, just exactly like it's supposed to.
Right.
The muscle forces blood out with absolute mechanical precision.
Mm.
But, and there's a massive catch here, the muscle wall has become so incredibly stiff that it cannot relax enough to actually fill back up with blood before the next beat.
Yeah, which is a huge problem.
Right. You have a pump that works, but a reservoir that's effectively frozen. This is a condition known as HFpEF, or heart failure with preserved ejection fraction. And it is, easily one of the most stubborn medical mysteries of our time.
It really is a phenomenal paradox and a devastating one for patients. Half of all heart failure patients actually present with this exact condition.
Wow, half.
Yeah, half. We are talking about millions of people globally who have a heart that, looks like it's contracting totally fine on an echocardiogram, but they are still suffocating because the plumbing just can't refill.
That's terrifying.
It is. And for decades, the medical community treated it as just a mechanical problem isolated to the heart muscle itself. But as we'll see today, HFpEF isn't just a heart muscle problem. It is a systemic whole body syndrome. It involves severe dysfunction in the microvasculature, which are the tiny blood vessels all over your body.
Which brings us to our actual mission for this deep dive. We are exploring a truly groundbreaking study from the journal Cell Stem Cell, published by Toyama and colleagues.
A fantastic paper.
Yeah, really incredible stuff. We're going to look at how these scientists literally built a beating mini heart in a dish to solve this HFpEF mystery.
Yeah.
And how they map the exact pathway a specific drug takes to fix this frozen pump.
To understand why this study is such a huge milestone, we kind of have to look at the historical failures of cardiovascular research.
Right.
Traditionally, we rely on animal models, specifically mice, to study heart disease. But mice are basically terrible at replicating HFpEF.
Why is that? Just because of the size?
Well, think about the biology. A mouse heart beats up to like 600 times a minute. Yeah, and a human heart beats maybe 70 times a minute. You simply cannot scale up the hemodynamics of a heart beating 10 times a second and expect the cellular physics of relaxation to perfectly mirror a human heart.
Because things are happening too fast.
Exactly. The timing of calcium channels, the protein recoil, it's all fundamentally different.
Which means we've been testing drugs on this alien system and wondering why they fail in human trials. That actually makes the 2022 FDA recommendation you mentioned in our notes a massive paradigm shift.
Oh, absolutely.
The FDA basically said, we recognize mice aren't miniature humans. They stated that animal testing is no longer strictly mandatory for drug approval if researchers can utilize what they call NAM's new approach methodologies.
Right, and that ruling fundamentally changed the landscape of biomedical research. This study by Toyama's team is just a premier execution of an M.
They skipped the mice completely.
Completely bypassed the mouse. They used human stem cells to model human heart failure, creating a bespoke testing ground that actually reflects our own biology.
Okay, let's unpack how they actually did this. Because the idea of just whipping up a human heart in a petri dish sounds, frankly, like pure science fiction.
It kind of does.
How do you go from a stem cell to a functional beating piece of tissue?
So the foundation relies on human-induced pluripotent stem cells, or hiPSCs. By taking adult cells and reprogramming them into this pluripotent state, the researchers basically had a blank canvas.
Okay.
From there, they cultivated a highly specific recipe of 3 distinct cell types to build their model. First, they grew cardiomyocytes, which are the actual contracting muscle cells.
Got it.
Second, epicardial cells, which form the outer structural layer of the heart tissue. And third, endothelial cells, which form the crucial inner lining of the blood vessels.
So they have the ingredients, the muscle, the wrapper, and the plumbing.
They took those 3 cell populations and mixed them into a biological scaffold made of porcine-derived collagen and fibrinogen.
Like a glue.
Exactly like a glue. It acts as the extracellular matrix to hold the tissue together. Then they cast this mixture into a tiny microscopic mold around 2 flexible, solid silicone posts.
Give you the structure.
Right. Those posts gave the cells something to anchor onto. And within just 1 week in the incubator, this chaotic mixture of cells literally self-organized into a cohesive tissue and started spontaneously beating.
That is wild. It's like creating a tiny biological rubber band strung between two posts, and it's just plucking itself over and over.
Yeah, that's a perfect visual for it.
And because it's hooked to these flexible silicone posts, scientists can track the exact physical deformation, right? They use motion imaging to measure how far the posts bend.
Which gives them the contraction force, the beating rate, and most importantly for this disease, the relaxation duration. They basically created a window into the exact mechanics of a living human heart, measurable down to the micron.
But I have to push back on this model for a second, though.
Sure.
I understand the FDA wants to move away from mice, but you can't just throw 3 types of stem cells into a dish with some collagen and, call it a human heart.
Fair point.
A real heart has a complex nervous system dictating its rhythm. It has changing systemic blood pressures. It has immune cells constantly circulating through it. Isn't a 3-cell model strung on a silicone post missing the forest for the trees a bit? Like, how does such a simplified system help us understand a complex whole-body syndrome like HFpEF?
That's the defining tension in in vitro research. You are totally right. It lacks the systemic nervous and circulatory inputs.
Right.
But that isolation is actually its greatest strength for this specific question. By stripping away all the systemic noise of the whole body, you isolate the local tissue environment.
Oh, I see.
If you can induce HFpEF in this isolated tissue, it proves that the disease is fundamentally driven by the local cells themselves, not just, external inputs from the brain or the kidneys.
It narrows down the suspects.
Exactly. But to your point, a healthy piece of engineered tissue, and HEHT, doesn't tell us much about disease. The researchers had to deliberately torture this tissue to induce the disease state.
Okay, so just to make sure we're keeping track here for the listener, so far we've established that HFpEF is a stiff heart that can't relax. Mice are basically useless for studying it because their hearts beat too fast. So scientists grew a 3D beating human heart tissue from stem cells to study it instead.
Yep, that's the setup.
So, audience, I want you to think about this. If you had a healthy, pristine mini heart beating away in a dish, what kind of biological junk food would you feed it to mimic human heart disease?
It's a great question.
So do they actually bombard it with?
They designed a very specific toxic soup to recreate the metabolic stress of a patient suffering from obesity, hypertension, and systemic inflammation.
Nasty stuff.
Very. The first ingredient was a massive dose of fatty acids. They used a lipid-rich protein called albumax to basically force the tissue into a state of severe metabolic overload.
Drowning the cells in fat. And the second ingredient?
A chemical inhibitor known as L-name.
L-name.
Right. The sole function of L-name is to block the production of nitric oxide, or NO. In a healthy body, your endothelial cells constantly secrete nitric oxide to signal the surrounding blood vessels and tissues to relax.
Okay.
By using L-name to sever that chemical signal, they artificially induced endothelial dysfunction.
So they soak this beating tissue in a high-fat diet and completely disable its ability to produce relaxation signals.
Yeah.
What does that actually do to the physical mechanics of the mini heart after, say, a couple of weeks?
The mechanical shift was undeniable. Under the imaging system, the contraction force remained completely preserved. The tissue squeezed just as hard as it did on day one.
What?
But the RDCD ratio, that's the ratio of relaxation duration to contraction duration, extended drastically.
It's essentially a rusty spring.
Oh, I like that.
Yeah, think about it. If you push on a heavy rusty spring, you can still compress it if you apply enough mechanical force. But the moment you let go, instead of instantly snapping back to its uncoiled resting state, it slowly, painfully grinds its way back. The contraction is preserved, but the relaxation is completely ruined.
That is exactly what's happening. And because they had this isolated model, they could basically slice that rusty spring open and look at the molecular rust under an electron microscope.
What did they find?
The structural damage inside the cells was profound. The mitochondria were completely collapsed. Their internal architectures were just distorted and choked with accumulated lipid droplets from the albumax they were fed.
Wow. They were physically suffocating under the lipid burden. Did this metabolic stress force the tissue to change its genetic programming too?
Completely. The tissue recognized it was failing and started broadcasting distress signals. It upregulated a gene called NPPB, which produces BNP.
Ah, right, BNP.
Yeah, for anyone who follows cardiology, BNP is the classic diagnostic marker. When a patient arrives at the ER gasping for air, the first blood test drawn is often for BNP to confirm heart failure.
So this little cluster of cells in a dish was screaming the exact same chemical warning as a human patient in the ER.
Precisely. They also detected massive spikes in IL-6, which is a primary driver of severe inflammation.
And what about the physical scaffolding itself? Like, if the relaxation time is increasing, something structural has to be stiffening the tissue.
It did. The tissue fundamentally rebuilt its own architecture to become more rigid. It stopped producing type 3 collagen.
Which is what?
Type 3 is the soft, pliable collagen that allows healthy tissue to easily expand. Instead, the tissue heavily upregulated type 1 collagen, which is dense, rigid, and meant for structural support.
So they essentially built a brick wall inside the heart muscle to cope with the stress.
Furthermore, they observed a really fascinating shift in a protein called titin. Yeah. Titin acts as the actual molecular spring inside the muscle sarcomeres. Healthy human hearts utilize a long, highly elastic version of this protein called the N2BA isoform.
Okay.
But under the stress of the high-fat, low-NO diet, the tissue's genetic splicing literally shifted to produce the N2B isoform. That version is much shorter and far stiffer.
That's crazy. So they didn't just add rust, they literally went in on a genetic level and swapped out the elastic springs for rigid ones.
Yes.
Okay, let's do another quick recap. High fat plus blocked nitric oxide equals a stiff, inflamed heart model. And this model perfectly matches the human disease, high BMP, high IL-6, stiff collagen. The contraction is fine, but it can't relax. They have successfully built a perfect physical replica of human HFpEF in a dish.
It's an incredible achievement.
But here's where it gets really interesting. Where do they even begin to find a drug to reverse that kind of structural damage?
Well, this is where the sheer utility of the new approach methodology really shines. Because they had a reliable, scalable model of a sick heart, they could just throw a massive library of existing cardiovascular drugs at it to see if anything could move the needle.
Just rapid-fire testing.
Exactly. They tested ARNIs, which are standard heart failure mainstays. They tested SGC activators, PDE5 inhibitors, even HCN inhibitors designed to alter heart rate.
And I imagine most of them did absolutely nothing for the relaxation duration.
Out of all the compounds tested, only one successfully normalized the relaxation time without suppressing the contraction force. It was a class of drugs known as SGLT2 inhibitors, specifically a drug called empagliflozin.
Wait, I need to pause right there. SGLT2 inhibitors are diabetes drugs.
They are.
Their primary mechanism of action is in the kidneys, right? They block the reabsorption of glucose, so the patient literally urinates out excess blood sugar. Cardiologists have noticed recently that patients on these drugs seem to survive heart failure better, but it's always been kind of a mystery.
A huge mystery.
So how does a diabetes drug, a kidney drug that manages blood sugar, fix a rusty heart spring?
That has been the billion-dollar question in cardiology for years. Using this in vitro model, Toyama's team was finally able to map the precise molecular communication highway the drug uses, step by step.
Okay.
They discovered that the SGLT2 inhibitor acts as a potent anti-inflammatory agent that restores a critical sequence known as the ENO-S-NO-cGMP-PKG pathway.
Okay, we're going to need to trace that highway very carefully because that is a serious amount of alphabet soup.
It is a lot, I know.
Where does this communication start?
It originates in the endothelial cells, remember, that's the plumbing layer.
Right.
Inside those blood vessel cells is an enzyme called enos. Its entire biological purpose is to manufacture NO, nitric oxide.
Got it.
Because nitric oxide is a gas, it easily diffuses out of the endothelial cell and travels across to the neighboring cardiomyocyte, the muscle cell.
Okay, so the blood vessel creates the nitric oxide gas and sends it over to the muscle cell. What happens when it arrives?
Once inside the muscle cell, the nitric oxide triggers the production of a secondary messenger called cGMP.
Okay.
You can think of cGMP as the internal alarm clock of the cell. As cGMP levels accumulate, it wakes up the ultimate authority in the sequence, protein kinase G, or PKG.
Right, PKG.
When PKG is activated, it basically phosphorylates the stiff titin proteins and the surrounding machinery, commanding the entire muscle apparatus to let go and relax.
Okay, that is a beautifully elegant chain of command. The blood vessel makes NO. The NO travels to the muscle. The NO triggers cGMP. The cGMP activates PKG, and PKG says, relax.
It really is elegant. But remember what the toxic disease diet did. The high fat and inflammation completely shut down the ENO-S enzyme in the blood vessels.
Oh, so the very first step was broken.
Exactly. The signal never even reached the muscle. The researchers found that the SGLT2 inhibitor repairs this by triggering AMPK phosphorylation.
Meaning what exactly? What is AMPK phosphorylation doing in this context?
Think of phosphorylation as attaching a chemical tag that acts like an ignition switch. The drug triggers this AMPK tag to attach to the dormant ENOS enzyme. It physically forces the enzyme to turn back on, jump-starting the production of nitric oxide and reopening the entire communication highway.
That makes so much sense. But if inflammation is what breaks this ENOS pathway in the first place, is this an external attack from the immune system? Because earlier I challenged you on the simplicity of this tissue model, missing a systemic immune response.
Right, you did.
If you introduce circulating immune cells into this isolated dish, does it accelerate the stiffness?
They ran that exact experiment. They introduced macrophages, which are the immune cells that mediate inflammation into the mini-heart. When they added M1 macrophages, which drive aggressive inflammatory responses, the stiffness and the relaxation time got significantly worse.
Not surprising.
Right. And when they added M2A macrophages, which promote tissue repair, the tissue showed slight improvements.
So the immune system definitely plays a role in amplifying the damage.
It amplifies it, yes. But here is the critical takeaway. Even when they completely scrubbed all macrophages from the tissue, when it was just the isolated muscle and vascular cells, the model still developed severe HFpEF when exposed to the high-fat diet.
Wow. Meaning the disease doesn't strictly require an external immune system attack to initiate.
Exactly.
The local cells in the heart tissue are fundamentally broken and driving their own localized inflammation.
The pathology is deeply embedded in the local tissue environment, and this finding flips our conventional understanding of heart disease on its head. When we discuss heart failure, the cardiologist's focus is almost always on the cardiomyocyte, the muscle cell.
Right, the heart muscle.
We treat it as the main character. But this study reveals a massive plot twist. What's fascinating here is the true hero of the story isn't the muscle cell at all.
It's the endothelial cell.
The real target of the disease and the indispensable middleman for the cure is the endothelial cell.
And the way they prove this causality is just staggering.
It's brilliant.
They build a brand new batch of mini-hearts. But this time, they intentionally left out the endothelial cells. They built a heart using only the cardiomyocytes and the structural epicardial cells. 0% blood vessel lining.
Just muscle and structure.
Right. And when they induced the disease in this tissue and applied the SGLT2 inhibitor, our miracle diabetes drug, the drug completely failed. It did absolutely nothing to fix the relaxation duration.
The drug was powerless because it doesn't communicate directly with the muscle. It strictly requires the endothelial cells to process the signal and produce the nitric oxide.
But they didn't even stop there. Just leaving the cells out might disrupt the structural integrity of the tissue, which could, skew the results. So they proved it genetically.
They utilized CRISPR gene editing technology. They took a population of perfectly healthy endothelial cells and selectively deleted the NOS3 gene.
What does that gene do?
That is the precise genetic sequence responsible for manufacturing the ENOS enzyme. So they essentially created mutant endothelial cells that looked structurally normal, but were genetically incapable of ever producing nitric oxide.
Oh, wow. So they built a new mini heart with these CRISPR-edited cells, exposed it to the toxic diet, added the SGLT2 inhibitor drug, and again, the drug failed.
It is an ironclad proof of causation. The diabetes drug protects the heart muscle by specifically rescuing the blood vessel cells.
But this brings up a foundational question. How did the endothelial cells get so damaged by the metabolic stress in the first place? Why does a high-fat diet cause them to shut down their nitric oxide factories?
To understand the root cause of the damage, we have to look at how cells manage their internal ionic gradients. It comes down to a phenomenon called ion overload.
Ion overload.
Yeah. Under the severe stress of the HFpEF conditions, the endothelial cells lose their ability to regulate their internal environment, and they become flooded with two specific ions: sodium and calcium.
They're just drowning in salt and calcium.
Precisely. In a healthy physiological state, cells utilize tiny transmembrane protein channels to pump these ions out against the gradient, maintaining a strict internal balance. Two of the most heavily relied upon channels are NHE, which exchanges internal hydrogen for external sodium, and NCX, which exchanges internal calcium for external sodium.
So these are basically little revolving doors for ions.
Yes, but under the stress of HFpEF, the regulation of these channels collapses. The doors get jammed open, and the cell is overwhelmed by toxic concentrations of sodium and calcium.
And I assume this massive ion flood is what triggers the severe internal inflammation that ultimately shuts down the ENOS enzyme.
Exactly. The cell prioritizes survival over communication, and the nitric oxide production halts entirely.
So how does our SGLT2 inhibitor actually intervene in this ionic flood?
The drug operates like a cellular bouncer at these specific membrane channels.
A bouncer.
Yeah. The researchers observed that the SGLT2 inhibitor physically suppresses the excessive expression of the NHE and NCX channels on the surface of the endothelial cells. By essentially boarding up these malfunctioning doors, the drug stops the unregulated influx of sodium and calcium.
Okay, let's do our final mini recap, if I have the full picture now. SGLT2 inhibitors cure the stiffness by restoring the NO highway. Endothelial cells, the blood vessel cells, are the crucial middlemen required for this to work. And the drug prevents the toxic sodium and calcium buildup by acting as a bouncer blocking the NHE and NCX cellular doors.
That is the complete, uninterrupted mechanical pathway of how this drug saves a failing heart, verified step by step in a human tissue model.
It's just amazing.
And if we connect this to the bigger picture, this research validates a monumental paradigm shift in cardiology. It strongly suggests that heart failure with preserved ejection fraction is not, at its core, a disease of the heart muscle. It is fundamentally a microvascular disease.
The tiny blood vessels get sick first.
They do. They lose their ability to regulate ions, they succumb to inflammation, and they stop sending out relaxation signals. The heart muscle isn't the instigator. It is merely a rigid, stiff victim of that microvascular silence.
It is a phenomenal journey through the microscopic mechanics of human biology. Thank you so much for acting as our guide through this incredibly dense and frankly fascinating landscape.
My pleasure.
The sheer reality that we are no longer guessing based on mouse heartbeats, but are actively mapping out molecular pathways on bespoke, lab-grown human tissues is just staggering. We are finding the exact cellular rust and designing the precise molecular intervention needed to dissolve it.
It is the realization of precision medicine. And, as powerful as these new approach methodologies are for cardiology, this raises a much larger question for us to consider as we step away from the data.
Oh. What's that?
Well, if endothelial cells, the delicate lining of the microvasculature, are the true key to this massive systemic heart disease, we have to remember that those exact same cells line every single capillary and blood vessel in our entire body. Right. They are the gatekeepers in our kidneys, in our liver, deep within our brain. So it forces you to wonder, what other supposedly unsolvable chronic organ diseases out there are actually just microvascular communication breakdowns, silently waiting for the right drug to reopen the highway?
Wow. Now that is a profound thought for you to ponder. We spend so much time looking at the macro organs that we forget about the thousands of miles of communication highways keeping them alive.
Exactly.
Maybe take a moment today to look up new approach methodologies and see what other human tissues scientists are building in a dish right now. Until next time, keep questioning the mechanics of the world around you and join us for another deep dive.